Our next presentation of this morning's session is entitled Screening Peanut Recombinant Inbred Lines for Aflatoxin Contamination Using In Vitro Seed Colonization of Aspergillus flavus. And our presenter is Raegan Wiggins.
Can you see it? Okay. All right.
Yes.
So thanks everybody for joining me. Thanks for the introduction, Dr. Kimura. So my name is Raegan Wiggins. I'm a master's plant breeding student at the University of Georgia in Tifton. I'm really excited to talk about my research on aflatoxin in peanut. So if you joined the webinar on Tuesday, you kind of got a little taste of what's so important to research aflatoxin. So hopefully I can extend on what some of the speakers were talking about there and reiterate some of the key points on why it's so important to research. So I'm going to start with talking about how aflatoxin actually affects our economy and our health. So basically, just a little background on aflatoxin. It's produced by a fungus called Aspergillus, mainly flavus and parasiticus. The highest risk crops for contamination are corn and peanut. Peanut itself is a $1.2 billion industry in the United States. So peanut pods can become infected with the fungus pre-harvest in the soil or either post-harvest along different points in the peanut supply chain. If environmental conditions are right, the toxin will be produced on peanuts, and then humans can become exposed to aflatoxin in one of two different ways. They can get it directly from infected peanut products, or even animals can transfer a type of aflatoxin to us through meat and dairy products if they were fed infected peanut or cornmeal in their livestock feed. Exposure to aflatoxin leads to enhanced health risks like liver cancer, immune disorders, and growth stunting. This is mainly a problem in developing countries, but in developed countries, it's mainly a problem because it leads to these really big economic losses. So I'll talk a little bit about that. So going off of the health risk, it's estimated that 68% of the world is chronically exposed to aflatoxin in some form, not necessarily through food products. But because of this, aflatoxin causes an estimated 1/3 of the entire world's liver cancer cases. This classifies it as the single most carcinogenic substance we've actually ever discovered. So it's a huge problem. Severe exposure cases can lead to a condition called aflatoxicosis, which causes an enlarged and diseased liver. And even in the worst cases, it can cause death. So just as an example, in 2004, it was discovered that food products from a single batch of contaminated food products being sold in Kenyan markets led to over 100 directly attributed deaths due to aflatoxin. So this wasn't the first or last case of aflatoxin outbreak in developing countries, and so it's a really big problem, and studies by the World Health Organization show just how widespread the problem is, with contaminated food products being found on pretty much every continent around the globe. Aflatoxin poses a concern for developed countries for a different reason, because individual countries are responsible for managing aflatoxin with their own sets of regulations. Now, these regulations are really important considering the health risks related to aflatoxin, but that doesn't necessarily mean that the regulations don't come at a cost. In the US alone, crop loads contaminated with a concentration of aflatoxin higher than 20 parts per billion is considered unsafe. The required testing and crop losses and management procedures related to aflatoxin leads to an estimated $1 billion in losses just for the United States alone, if not more than that. So considering all these problems with aflatoxin, that makes it a really important subject to research. So we'll talk a little bit about our research objective. So our research goal is to try and help peanut in particular combat aflatoxin contamination. So we've hypothesized that aflatoxin resistance can segregate in a recombinant inbred line population, which I'll kind of describe what it is in just a few minutes, and we hypothesize that the resistance is controlled by QTL, which can be mapped and linked to specific markers in the Arachis genome. To test our hypothesis, we developed a population, we phenotyped it, and we're working towards a genetic analysis. So let's talk a little bit first about the population that we developed. So it's based on this study that really helped to motivate our parental selections. One genotype called ICG 1471 demonstrated some comparative resistance to aflatoxin in this study. So you can see the figure on the left shows the amount of fungal growth that was found on all of the tested genotypes, with no significant differences in growth detected. However, the middle figure shows the concentration of aflatoxin that was found on the tested genotypes. And you can see the ICG71, which I've boxed in here, has lower aflatoxin contamination compared to some of those other lines. This means that we have some sort of resistance in ICG 1471, which is really interesting. In addition, this study showed another genotype that groups out here called Florida 07, which has higher aflatoxin contamination and overall higher ratio of fungal growth to aflatoxin. So these two genotypes, which vary for our aflatoxin trait, they make the perfect combination actually for developing something called a recombinant inbred line population. This is because the progeny of these crosses, they'll have segments of genetic material from both of these parents, which will allow us to determine which specific material segments may be contributing to our aflatoxin resistance trait in this really special line, ICG 1471. So we can talk a little bit about how we designed our experiment around crossing these two parents. So after we crossed these and we self them, we started phenotyping for aflatoxin in the F6 generation. And we inoculated 4 replicates of seeds from each of 100 recombinant inbred lines. We controlled for things like fungal spore concentration in our inoculum, seed health, incubation temperature, and humidity through using humidity chambers. This way we can infect our seeds as evenly as possible. The inoculations were performed with a special strain of Aspergillus flavus, and so this strain actually produces a green fluorescent protein, and so you can see that kind of in this image. All of those lit up green areas are areas on our peanut seed coat where we have Aspergillus flavus growth, so this helped us to visualize our fungal growth. So after 7 days of incubation in humidity chambers, each of our reps of each of our lines were ready to test for aflatoxin contamination. And so we did this, and it's a really long procedure. We have to grind seeds for each replicate, and then we have to extract aflatoxin using a buffer solution. And then we have to put that solution through something that's called an immunoaffinity column. And then that solution can go into a fluorometer, and that will give us the aflatoxin concentration out. So then once we have all of this data, aflatoxin data on each of our lines, we decided to do further testing on some of the lines that had the lowest contamination values and the highest contamination values, and we tested those further after another generation of advancement in the F7 generation. So we actually ended up selecting 22 lines to continue testing on for 3 more replications of the experiment. So this is a little bit about our first data set, which were our F6 seeds. So you can see in the plate up there in the top right corner, that's what one of the plates would have looked like a week post-inoculation. So we did, so this plate has 6 lines on it, 1 replicate of each line. And so those lines are replicated within these 6 well plates. And so we tested 100 of these recombinant inbred lines. So so we selected 15 lines based on our aflatoxin data that had the lowest aflatoxin concentrations, and then we ended up selecting 7 lines that had the highest aflatoxin concentration values based on this F6 data. So we actually ended up selecting more low contamination lines than high contamination lines. And this was because we did 4 replicates of the experiment, and so we wanted to make sure that none of the seeds or none of the lines that we tested escaped contamination. And so we casted a wider net for our low contamination lines for additional retesting, just to make sure that we didn't have any falsely positive results. And so before I go on to talking a little bit about these 22 lines that we selected and what the F7 data looked like, before we went on to test our seeds in the F7 generation, we screened for another trait in these lines. And so these 2 parents, they differed for aflatoxin resistance, but they also differ for OL ratio, which is the ratio of oleic to linoleic acid in the seeds. And so this is a trait that has been implicated to affect aflatoxin production. And so we wanted to make sure that this trait wasn't the only thing that was contributing to the resistance we were seeing in our F6 seeds. And so we screened for these for FAD2A and FAD2B, which are these homologous genes that affect OL ratio in peanut. And so a high OL ratio is a favorable trait for commercial peanut varieties. But we wanted to make sure that we had some low contamination lines that would have the high OL trait. And so what we ended up finding out is we had, after we screened for OL ratio, is within our low contamination lines, we had 4 normal OL lines, and then we had 3 high OL lines, which was really good. And then in our high contamination lines, we had 3 that were high OL and 2 that were normal. And then all of the other lines that we tested were heterozygous, adding 1 or more of these loci. And so this was good because it means we could continue testing in the F7 and that we might have some other genetic mechanisms that were contributing to aflatoxin resistance, and it wasn't just contributing, the OL ratio wasn't the only thing that was contributing to it. So then we were able to collect our F7 seed data. And so what we did is when we retested these lines, these 22 lines, we increased temperature, which increased our disease pressure. This gave us some higher aflatoxin contamination values for these lines. And so for these 3 more replicates, we had some really, really high contamination values. This made it kind of difficult to do the analysis just because we had some lines that were testing as low as 1 or 2 parts per billion for aflatoxin, and we had some lines that tested close to 1 million parts per billion for aflatoxin. And so that large variance in the data set made it really hard to analyze, but we ended up running an ANOVA on the combined data, and then we used an LSD test, which was a less conservative mean test that allowed us to analyze the data a little better. We ended up finding, actually parsing out 4 of the lines that were consecutively low contamination lines. And so you can see those pictured here. I really love taking pictures of my baby peanuts. So I take them from the time that they're babies as seeds and all the way until they have their own kids whenever we turn them up in the field. So all of our MVP peanuts are pictured here. We actually ended up extracting DNA from these 4 lines along with 4 lines that were high aflatoxin contamination. And so we're using these for genetic analyses. And so it was 4 lines out of the 15 we originally selected for that we extracted data from. And just to kind of summarize, because that was kind of a lot of dense information, I ended up phenotyping 100 F6 lines from this recombinant inbred line population for aflatoxin contamination. We narrowed that down to 22 lines that we tested in F7. We actually screened all of those 22 lines for F82A and F82B. And then I analyzed the data and we identified 4 low contamination lines, and we extracted DNA from those and the high contamination lines. And we're planning on using a technique called QTLC to analyze our data. And so we're hoping that we can identify some genetic regions that are controlling resistance. It's really great that we have a published peanut genome now, because we can use the resulting genetic data to actually maybe in the future develop markers for aflatoxin resistance. And this is a really, really big deal, because if we do have a validated marker for aflatoxin resistance, it can be integrated into marker-assisted breeding programs, and that can save the money and lives of breeders literally all over the world. And so it's really exciting research, and I've been really fortunate to work on it. And so with all of that, I want to acknowledge everybody who made this research possible. Of course, my lab, the Raegan's lab in Tifton, Georgia, along with my committee members, Dr. Clevenger, Holbrook, and Jackson, the USDA, which allowed me to use some of their equipment for my bioassays, along with the Peanut Foundation and Morris Ridley for funding my research. And so with that, I will take questions.
Thank you for the presentation. We have time for questions. And the first question comes from Dr. Dan Anko at Clemson University. He says, nice work. According to the middle graph on the population background slide, how did ICG 1471 have less than 0 parts per billion aflatoxin concentration? Possible innocent artifact of axis labeling.
Oh, sorry, yeah, that probably is the access labeling. It did have some aflatoxin contamination in the study. Sorry about that.
Okay. Second question would be, if you were to look at the lines that had the low aflatoxin contamination, can you suggest some similarity between them, or suggest some mechanism that might lead those lines to be more favorable in reducing aflatoxin than those which had high aflatoxin? Is there some characteristic that may explain or help to understand why this happens?
Yeah, so actually, I went back and I looked at some of those 4 lines in particular. I looked at some of the seed images. I looked at the above ground plant images, the below ground plant images, and they're actually all pretty different. They've got a variety of seed coat colors, so that wasn't really something that I saw that was affecting it. There's been some RNA-seq studies on aflatoxin that have proposed like certain genes that are affecting aflatoxin concentration, but that would be something that you would have to do more work on with these lines in particular. So there's nothing really for these 4 lines that I could propose, but I definitely think it's going to be interesting to see the genetic data that comes out of this.
Okay, thank you, and that's the time we have for questions. Thank you for your presentation.